Although the axolotl is colloquially known as a walking fish, it is not a fish, but an amphibian. The species was originally found in several lakes, such as Lake Xochimilco underlying Mexico City. Axolotls are unusual among amphibians in that they reach adulthood without undergoing metamorphosis. Instead of developing lungs and taking to the land, adults remain aquatic and gilled. Axolotls should not be confused with water dogs, the larval stage of the closely related tiger salamanders, A. tigrinum and A. mevortium, which are widespread in much of North America and occasionally become neotenic. Neither should they be confused with mud puppies, Nectaris spp, fully aquatic salamanders that are not closely related to the axolotl but bear a superficial resemblance. As of 2010, wild axolotls were near extinction due to urbanization in Mexico City and consequent water pollution, as well as the introduction of invasive species such as tilapia and perch. They are currently listed by CITES as an endangered species and by IUCN as critically endangered in the wild, with a decreasing population. Axolotls are used extensively in scientific research due to their ability to regenerate limbs. Axolotls were also sold as food in Mexican markets and were a staple in the Aztec diet. Surveys in 1998, 2003, and 2008 found 6,000, 1,000, and 100 axolotls per square kilometer in its Lake Xochimilco habitat, respectively. A 4-month-long search in 2013, however, turned up no surviving individuals in the wild. Just a month later, 2 wild ones were spotted in a network of canals leading from Xochimilco. The city is working on conserving axolotls by building axolotl shelters and conserving remaining and potential habitats for the salamanders. A sexually mature adult axolotl, at age 18 to 24 months, ranges in length from 15 to 45 centimeters, 6 to 18 in, although a size close to 23 centimeters, 9 in, is most common and greater than 30 centimeters, 12 in, is rare. Axolotls possess features typical of salamander larvae, including external gills and a caudal fin extending from behind the head to the vent. Their heads are wide, and their eyes are lidless. Their limbs are underdeveloped and possess long, thin digits. Males are identified by their swollen cloacae lined with papillae, while females are noticeable for their wider bodies full of eggs. 3 pairs of external gill stalks, rami, originate behind their heads and are used to move oxygenated water. The external gill rami are lined with filaments, fimbriae, to increase surface area for gas exchange. 4 gill slits lined with gill rakers are hidden underneath the external gills. Axolotls have barely visible vestigial teeth, which would have developed during metamorphosis. The primary method of feeding is by suction, during which their rakers interlock to close the gill slits. External gills are used for respiration, although buccal pumping, gulping air from the surface, may also be used to provide oxygen to their lungs. Axolotls have 4 pigmentation genes. When mutated, they create different color variants. The normal wild-type animal is brown, tan with gold speckles and an olive undertone. The 4 mutant colors are leucistic, pale pink with black eyes, albino, golden with gold eyes, axanthic, gray with black eyes, and melanoid, all black with no gold speckling or olive tone. In addition, there is wide individual variability in the size, frequency, and intensity of the gold speckling and at least one variant that develops a black and white piebald appearance on reaching maturity. Because pet breeders frequently cross the variant colors, animals that are double recessive mutants are common in the pet trade, especially white-pink animals with pink eyes that are double homozygous mutants for both the albino and leucistic trait. Axolotls also have some limited ability to alter their color to provide better camouflage by changing the relative size and thickness of their melanophores. Habitat and ecology. Axolotl in captivity. The axolotl is only native to Lake Xochimilco and Lake Chalco in the Valley of Mexico. Lake Chalco no longer exists, having been drained as a flood control measure, and Lake Xochimilco remains a remnant of its former self, existing mainly as canals. The water temperature in Xochimilco rarely rises above 20 degrees Celsius (68 degrees Fahrenheit), though it may fall to 6 to 7 degrees Celsius in the winter, and perhaps lower. The wild population has been put under heavy pressure by the growth of Mexico City. The axolotl is currently on the International Union for Conservation of Nature's annual Red List of Threatened Species. Non-native fish, such as African tilapia and Asian carp, have also recently been introduced to the waters. These new fish have been eating the axolotl's young, as well as its primary source of food. Axolotls are members of the tiger salamander, or Ambystoma tigrinum, species complex, along with all other Mexican species of Ambystoma. Their habitat is like that of most neotenic species, a high-altitude body of water surrounded by a risky terrestrial environment. These conditions are thought to favor neoteny. However, a terrestrial population of Mexican tiger salamanders occupies and breeds in the axolotl's habitat. The axolotl is carnivorous, consuming small prey such as worms, insects, and small fish in the wild. Axolotls locate food by smell, and will snap at any potential meal, sucking the food into their stomachs with vacuum force. 6 adult axolotls, including a leucistic specimen, were shipped from Mexico City to the Jardin des Plantes in Paris in 1863. Unaware of their neoteny, Auguste Dumeril was surprised when, instead of the axolotl, he found in the vivarium a new species, similar to the salamander. This discovery was the starting point of research about neoteny. It is not certain that Ambystoma velici specimens were not included in the original shipment. Willem Lofberger in Prague used thyroid hormone injections to induce an axolotl to grow into a terrestrial adult salamander. The experiment was repeated by Englishman Julian Huxley, who was unaware the experiment had already been done, using ground thyroids. Since then, experiments have been done often with injections of iodine or various thyroid hormones used to induce metamorphosis. Today, the axolotl is still used in research as a model organism, and large numbers are bred in captivity. They are especially easy to breed compared to other salamanders in their family, which are almost never captive bred due to the demands of terrestrial life. One attractive feature for research is the large and easily manipulated embryo, which allows viewing of the full development of a vertebrate. Axolotls are used in heart defect studies due to the presence of a mutant gene that causes heart failure in embryos. Since the embryos survive almost to hatching with no heart function, the defect is very observable. The axolotl is also considered an ideal animal model for the study of neural tube closure due to the similarities between human and axolotl neural plate and tube formation. The axolotl's neural tube, unlike the frog's, is not hidden under a layer of superficial epithelium. There are also mutations affecting other organ systems, some of which are not well characterized and others that are. The genetics of the color variants of the axolotl have also been widely studied. Regeneration. The feature of the salamander that attracts most attention is its healing ability. The axolotl does not heal by scarring and is capable of the regeneration of entire lost appendages in a period of months, and, in certain cases, more vital structures, such as tail, limb, central nervous system, and tissues of the eye and heart. Some have indeed been found restoring the less vital parts of their brains. They can also readily accept transplants from other individuals, including eyes and parts of the brain, restoring these alien organs to full functionality. In some cases, axolotls have been known to repair a damaged limb, as well as regenerating an additional one, ending up with an extra appendage that makes them attractive to pet owners as a novelty. In metamorphosed individuals, however, the ability to regenerate is greatly diminished. The axolotl is therefore used as a model for the development of limbs in vertebrates. Genome. The 32 billion base pair long sequence of the axolotl's genome was published in 2018 and is the largest animal genome completed so far. It revealed species-specific genetic pathways that may be responsible for limb regeneration. Although the axolotl genome is about 10 times as large as the human genome, it encodes a similar number of proteins, namely 23,251. The human genome encodes about 20,000 proteins. The size difference is mostly explained by a large fraction of repetitive sequences, but such repeated elements also contribute to increased median intron sizes, 22,759 bp, which are 13, 16 and 25 times that observed in human, 1,750 bp, mouse, 1,469 bp, and Tibetan frog, 900